Hello and welcome to your lab midterm. This is what we went over. The parts of a scientific method, there's the observation and your question. Then you go over the research background, you state a hypothesis, then you design and test an experiment. You then analyze your data, and then you report your results. The difference between plants and animal cells is that plant cells are freaking square-ish. They have cell walls, and they've got chloroplasts, where animal cells have neither. When you read a scientific article, there are different parts. The first part is the abstract, which is a summary of your results. Then you have your introduction, which states your purpose and the hypothesis. Then it goes into the methodology, which pretty much explains how and when and what you did to get your experiment done and blah. Then you have your results, in which you state a summary of your data and what you saw. And then you have your discussion. This is where you interpret the data, you state whether or not your hypothesis was correct. You compare your conclusion to the conclusions of similar tests, and you state where you could have messed up and what could have been done to make it better, and you state for further testing what can be changed to the experiment to get better results. Excuse me. Then we went over and then you state you make your acknowledgments, thanks the buggers who helped you out, and then you have your works cited. Then on to calorimetry, whatever. There's Beer's law, which pretty much talks about absorbance concentration, and in class we measured the absorbance and calculated the concentration. To do this, you take your absorbance, which equals molar extinction coefficient, times the distance light passes through a solution, times the concentration of the absorbing compound in, this is important, moles over liters. And then you guys got to know the parts of the spectrometer, which in my opinion is a machine you use to measure ghosts. Get it? What you guys need to know is that the left knob, this is my left, you set the absorbance to infinity. The right knob, this is my right, you set the absorbance to 0. 0. And then you guys need to know about the macromolecules. The basic units of each macromolecule we'll go over individually. First, there are carbohydrates, which are made out of carbons, hydrogens, and oxygens. Excuse me. They build into mono, di, and polysaccharides, and also into starches. Then there are the lipids, which are also made out of carbon, hydrogen, and oxygen. These make up fats, oils, waxes, fats. Excuse me again, I'm having tea. And these fats, oils, and waxes can be unsaturated or saturated depending on which one you make. Lipids can also form phospholipids and steroids. Excuse me. Then there are proteins, which are made out of polypeptides, which are chains of amino acids. They come in primary structures, which are the number and sequence of the amino acids. Secondary structures, which are the organizations of said amino acids. Tertiary structure, which is the folding of the proteins. And the quaternary structure, which is the link between the polypeptides. And finally, there are nucleic acids, which are made out of nucleic chains. In class, we then took a reagent and used it to test for each macromolecule. You used the Benedict's test to test for simple sugars. You used Lugel's test to test for starches. You used the Sudan 4 Sudan 4, you test for lipids, and you use the Barrett test for proteins. When we went over enzymes in class, we used acid phosphate, and phosphate is catalyzed by acid phosphate. Duh! What an enzyme does is it speeds up a reaction. Then you have to know why leaves are green. Do I really even have to go over this? I guess I do. Leaves are green because they reflect green light and absorb all the other colors. Okay? Understand? Reflect green, absorb everything else. Fixed carbon, this means that it's going from an inorganic to an organic. The photosynthesis reaction is CO2 plus water produces, with the help of solar energy, a sugar of sorts plus oxygen. Long wavelengths of light also have less energy than short wavelengths. I'd go into more detail, but I'm bored. And finally, the difference between a polar and a nonpolar molecule is the molecule shape. Well, kiddos, that is all for this session of helping you study. Bye-bye.